Good afternoon, everyone. Welcome to Science Says. My name is Tom Lynch. I'm the president and director of the Fred Hutch, and I just could not be happier to be with all of you today. It's Giving Tuesday, and we are so grateful for everyone who fuels progress at the Fred Hutch, starting with all of you who have given to the Hutch and helped us get to where we are. I just returned from a meeting in New York City where I got to meet members of the Rubin family and the Rona Jaffe Foundation, who funded endowed chairs for work in cancer, in bone marrow transplantation. We had 4 bone marrow transplant survivors, all greater than 25 years, who were at the dinner. And when you see that impact of the science at the Fred Hutch and how it turns into impacts for patients, it's incredibly powerful. And that's what we're going to talk about today. I want to thank our presenting sponsor, Microsoft. Microsoft employees support the Hutch in so many ways, whether we're talking about technical support or we're talking about ways of accessing the cloud or donations from Microsoft employees. It's an incredibly generous community. In fiscal year 2022, Microsoft employees donated $255 million and volunteered more than 750,000 hours across the world to various different charitable causes. So Microsoft, thank you very much for helping. So one of the things I've learned from talking to donors and people who support us is I have found that the best way to match an interest of the donor with an event or a group at the Hutch is really to focus on what is it that's motivating the donor, what motivates the person to give. Why do you give to one foundation and not another? And when I talk to people about cancer and viral disease, I'm incredibly impressed by how important the donor community values collaboration. As many of you know, we just were very fortunate, extremely fortunate, to receive several very large and impactful gifts from Mike and Jackie Bezos, from Stuart Sloan and Molly Nordstrom, that have made an enormous difference to what we can do at the Fred Hutch. And in speaking with both of those groups of supporters, the concept of collaboration came over time and time again. They want to know, and you want to know, that the science we're doing here doesn't just stay at the Fred Hutch, that it involves other people, other teams throughout the country, throughout the Hutch, throughout the University of Washington to hit other different parts. And the Hutch is an extremely collaborative place. We coordinate national and international government-funded collaboratives like the Women's Health Initiative, the HIV Vaccine Trials Network, and the COVID Prevention Network. Excuse me. Our investigators and labs partner with peers at other institutions, Memorial Sloan Kettering, Mass General, Dana-Farber, University of Texas, UCLA, to work in cancer and in viral diseases. We work with pharmaceutical and biotech companies to help breakthrough discoveries reach the market where they can impact patients. We partner with patients and families who trust their care to us and choose us to be able to treat their most valued people in their lives. We work closely with schools and teachers, and we're going to talk about some of that today and the impact we've made in education at the Fred Hutch. And so we convene peers often to share information, collaborate within our own walls, but also to create a community and a culture of science in these areas. So today we're going to focus on 3 collaborations that inspire us. I hope they're going to inspire you too. You're going to meet a scientist and a surgeon who are working together to understand the seeds of breast cancer metastasis. You're going to meet a researcher who launched one of the very first longitudinal studies of COVID-19 in March of 2020, and a firefighter who volunteered to be part of that study. You're going to meet a genetics researcher and an aspiring physician scientist who met through our summer high school internship program and are still working together on cures for children with leukemia. Great examples of collaboration. So I'd like to bring on our first pair. And they may look a little bit familiar because both Dr. Javed and Dr. Gahar are on our new ad campaign, which is airing in Washington State, and you may see them there. If you haven't seen them there, it's great that you're going to have a chance to see them today. Dr. Sarah Javed leads the breast health clinic at Fred Hutch and is chief of breast surgery at UW Medicine. Dr. Cyrus Gajar founded and leads the laboratory for the study of metastatic microenvironments, where he studies why dormant breast cancer cells that have traveled to distant organs sometimes reactivate years or decades after a patient's initial treatment, causing metastatic disease. Together, they design and lead a clinical study called the DUET study that aims to understand how cancer cells that have migrated from the breast to the bone marrow manipulate the environment of the bone marrow and the immune system in order to survive. And we really hope that these studies are going to be impactful for patients. So Cyrus and Sarah, welcome so much. So I'm going to start with Sarah. And the question for you, Sarah. Sarah, in my practice, I was a clinical lung cancer physician. I took care of lots of patients with lung cancer in my career. And I'll never forget being sent a patient, and it would probably happen once a year. I had a very big practice. And I would get a patient sent to me who had a pleural effusion. And for those of you who aren't physicians, a pleural effusion is fluid that is in between the lung itself and the chest wall. It accumulates and it makes people short of breath. Well, most of the time that you get a pleural effusion, it turns out it's probably lung cancer. Not always, there can be other things, but it can be lung cancer. And they sent a patient to me, and I said this would happen every year or so. Sent a patient to me with a big pleural effusion, and we drained the fluid, and the pathologist called me back a week later, and they said, Tom, this is breast cancer. And I would then find out that the patient had a history of breast cancer 18 years ago, 25 years ago, 7 years ago, years and years later. The patient had thought they were cured of their disease, and yet it comes back as a pleural effusion, usually estrogen receptor positive, were the patients who I tended to see just because of the way it worked. But so, Sarah, tell us about that phenomena. Is it unusual? Does it happen? And why does it happen?
Thank you, Tom. Thanks for the lovely introduction as well, and for inviting us to take part in this today. I think, the particular disease type that you speak about, hormone receptor positive breast cancer, can often have a more indolent or less aggressive course. And I think we are more likely to see these cells remain dormant distantly and exhibit or demonstrate long-term distant recurrences. We speak about how recurrences are most common within the first 5 years of diagnosis, but it really varies drastically depending upon the histologic or disease subtype. For example, hormone receptor negative cancers we call triple negative cancer, or HER2 positive breast cancer, more unusual or rare types, are more prone to recurring within the first 1 to 2 years after completing treatment, where these estrogen receptor positive can pop up years later, distantly. I wish I knew why, and I think that's what Cyrus and I are aiming to study here in our DUET work.
And so, Cyrus, tell me where you are. What are you understanding? So when I tell you about these patients, what bells go off in your head in terms of thinking about the biology of why this might be happening?
Yeah, well, thank you again for having me on, Tom. And it's wonderful to be paired with Sarah again. I think the important thing for the audience to understand is if breast cancer ended in the breast, then we would be in great shape. We have amazing surgeons like Sarah who are able to remove tumors and ensure that they don't come back. And we have a battery of therapies that we can throw at those tumors, including radiation, to ensure that those tumors don't come back in the breast. But what happens all too often is that you have these recurrences at distant sites like the liver, the bone marrow, the brain, and the lung. And when they come back, they destroy these critical organs, and that is often, if not always, lethal. So as Tom mentioned, as Sarah mentioned, this can happen 5, 10, 15, even 30 years after the initial diagnosis. And what we know is that these cells have left the breast tumor cells have left the breast. They're still breast tumor cells, and they're sitting in tissues like the lung, the bone marrow, the brain. And what we know is that they've set up shop around the vasculature. So they take the vasculature like highways to get to these distant organs. They exit, but they don't go far from that highway. They basically sit right on the surface of blood vessels. And this niche, or this microenvironment called the perivascular niche, is really important to regulating these cells, to keeping them dormant, to helping them survive chemotherapies, and to helping them evade the immune system. So these are the critical things that we know, and we've found this in models, and we've studied this in models. And now we really need to know what we've discovered in all of our models that we use, does it apply to people? And so that's really where this duet study comes in, that Sarah and I and a number of other people at Hutch and also at other institutions around the country are working on, is to understand, to actually purify and isolate these cells from people, to study them, to profile the tumor cells, the dormant cells themselves, as well as their microenvironments, and understand if all these things that we're discovering in different models in the lab apply to humans, and are the vulnerabilities that we're discovering going to be able to be taken advantage of in humans so that we can deprive these cells of survival cues, eliminate them from the equation, and stop patients from looking over their shoulders and worrying about recurrence 10, 20 years down the road.
And so, Cyrus, I'm just going to follow that up. What's the trigger, though, that sets it off? Meaning the patient I described who had the 18 years in between the two things, why was it 18 years and not 14 years or 30 years? What trigger caused it to start causing a pleural effusion or to go from the bone marrow to the pleural space? What was it? What's that trigger?
Yeah, it's a little bit of a cop-out answer, but the answer is that it can be so many different things. And I think the key is tissue homeostasis. Maintaining homeostasis, or that balance within a tissue, is really critical to keeping this cell in a dormant state. So what we know, as I had mentioned before, these cells sit on the endothelium or the vasculature, the blood vessels within an organ. If you disrupt the blood vessel that a tumor cell is sitting on and cause it to sprout, this process called angiogenesis, the tip cells that actually lead that sprout lose all of the suppressive cues that are present around the vasculature and secrete a bunch of pro-growth signals that can trigger these cells actually to wake up. If you have inflammation, and I'm talking about, pretty profound, large-scale inflammation, and it happens in the right time and place, it can also trigger these cells to wake up. But there's an interesting component to that, in that all of these dormant cells are not capable of waking up. And there's something that happens over time that I believe causes less and less of these cells to be even capable of responding even to these pro-inflammatory cues or these cues that come with loss of tissue homeostasis. So there's something about cells that remain receptive that we have yet to understand, that we need to be able to define. And there's something about these instances, these triggers occurring in the right time and place to the right cell that then allow it to wake up. And these are the kind of things that we still can kind of hand wave explanations for, but until we really study this in people, we're not going to be able to give an answer.
So Sarah, tell me a little about what exactly the duet study is. What exactly are you and Cyrus trying to figure out?
We're trying to be able to predict in the future which patient's disease type, what factors are predictive of distant metastatic recurrence based upon looking at, as Sarah said, the kind of the microenvironment within the bone marrow. And what it involves is we're actually enrolling patients not with metastatic disease, but with early stage invasive breast cancer, stage 1 through 3, at the time of diagnosis before they've received any therapy, including surgery. So we enroll them very early on in their course. And at the time of enrollment, we're not only drawing a blood sample, but we're also doing in the operating room while they're under anesthesia, a bone marrow biopsy as well as a bone marrow aspirate. Cyrus is bringing those to the lab and analyzing those to look at kind of markers for progression or distant metastatic disease. We're following these patients out for 9 to 10 years to look at that.
And so, Sarah, if you looked at patients who had early stage breast cancer in the bone marrow, how often do you find clinically recognizable occult metastasis in the bone marrow if you look carefully?
Well, we don't currently look for it, but Cyrus, maybe you can speak to that.
Yeah, so the studies that have been done historically bin down a maximum of 2 million cells from the bone marrow on slides and then stain for a marker of breast cancer cells. And they identify at least one cell in about 30% of all early stage breast cancer patients. If one of those cells is present, that patient has about a 3-fold higher risk of recurrence over the next 5 years than the patient where you can't find that cell. What we're doing that's a little bit different is going far deeper, but going about an order of magnitude deeper. We're profiling and looking through 20 million bone marrow cells to then purify disseminated tumor cells and then sequence and study them. So it's a little bit, it's deeper than what's been done before, and so we're going to maybe come up with different answers than what have been uncovered before.
So the last question, and you can collaborate on the answer if you like. What caused you to work together? Tell us how that actually happened. Who made the first phone call? How did you decide to work on this project? Sarah, you've got so much surgery to do. You could just do surgery all day long. And Cyrus, your lab is enormous. You've got tons of postdocs and graduate students to mentor. What brought you together? Where was the spark? What was the idea? How did the collaboration happen?
So I think when you enter academic medicine, this is what drives you. You really want to not only practice surgery and take care of patients, but you want to advance the specialty and the discipline, and that really takes partnership. As a surgeon, you hit the nail on the head. I'm in the operating room most days. I can't dedicate the time, nor do I have the training and background and expertise like scientists like Cyrus. And so it really does take a partnership. I think we first met probably, at least 5 to 7 years ago. It was, we have these breast cancer seminar series. We have breast faculty retreats where we present, developing technologies as well as science in our program, not only medical oncology, surgical oncology. Our researchers are present for that. So I think that's where we met and decided that we should partner up. And as the head of breast surgery at UW, it made sense for me to recruit my colleagues and our program patients to enroll.
Terrific. So Sarah and Cyrus, we're going to be back in just a few minutes for the Q&A with everyone. I encourage everyone to put your questions into the Q&A, whether they're about breast cancer, or whether they're about COVID, whether they're about the Hutch. Please put your questions, and during the Q&A session, we'll have Cyrus and Sarah back, among other people, to be able to answer some of these questions. Now I'd like to introduce our next duo. Julie Sartosky is a nurse practitioner with the Seattle Vaccine Trials Unit, which is based here at the Fred Hutch. Welcome, Julie. At the start of the pandemic, she started recruiting healthcare workers and first responders into something called the Seattle COVID Cohort Study. Understand how the immune system interacts with the virus. And you have to remember, when that was happening, we didn't know much about the virus. We didn't know how it spread. We were nervous and scared. And we knew Seattle was one of the epicenters of where it was starting. And Julie's study was one of the first to get going in that area. And she met at that point Mike O'Brien. And Mike is a captain with the East Side Fire and Rescue, stepped up right away to volunteer for this study. And nearly 3 years later, he's still part of it. And Mike is joining us from his remote location over on the East Side. Mike, it's terrific to have you with us. Julie, it's great to have you as well. Thank you so much. So Julie, I'm going to ask you the first question, which is, tell us about the study and what have you learned to date? What's the most compelling thing you've learned to date about your study?
Well, as you mentioned, the study started at the beginning of the pandemic. Since that time, we've enrolled over 600 participants. And really in the beginning, it was all about learning what is going on with this new virus. How does it work in our immune? And at the same time, first responders weren't able to get tested. As you may recall, they were out in the community, but nobody could get tested. So we thought, let's work together. Let's offer testing, look in the blood for antibodies, do nasal swabs, and also at the same time, we'll be studying the virus. And so that's how we started. And we also enrolled people in the study who were ill with COVID, and we have been following them since the beginning as well. And I think the thing that really stands out to me is our journey with long COVID and how that has evolved over time. So I remember a month after the study started, there was one day where like 4 different people came back for a visit after a month and they weren't getting better, or they had these new symptoms that were odd. And I was like, this is different. This isn't what I was expecting. And of course, that was the beginning of this thing we now know as long COVID. And it turns out that about 40% of that original cohort went on to have long COVID. And in looking at those folks, we have found that there's some differences in their blood. And one of the things that we've seen is that their inflammatory response or inflammation doesn't shut down properly. And so that's something we're looking into further. Is that a route that we can use to treat long COVID?
So, Mike, one thing you may not know is that the head of our vaccine unit here at the Fred Hutch is Julie McElrath, and Julie's son is a Seattle firefighter. So firemen always had an inside route at the Fred Hutch because the head of the vaccine study is very partial to firefighters. So it's terrific that first responders were the focus of this initial study. And so, Mike, I guess the question for you is, it takes a lot of courage to volunteer to be on a study. You don't know exactly what's going to happen from it. Tell us a little bit about what went through your mind and what it's been like to be part of this trial.
Sure. So I've been a first responder since 2000, and in that amount of time, we've prepared for, I believe it's 4 emerging respiratory syndromes and Ebola. And so we've always been having memos, conversations, practical and physical training, PPE, engineering controls about how to prepare and respond to these different emerging viruses. And we've never had one come through Seattle the way COVID did. So when the opportunity, alerted via an email, and an opportunity came out to be part of a study that would provide us more information on what we're doing right, what we're doing wrong, access to testing and lab work, and help an organization like Fred Hutch put out accurate information about what we're seeing and what we need to do in the future to keep all the first responders safe. It was a no-brainer to sign up for being part of this study.
So, Mike, another question I would have for you, as a physician and our nurses here at the Hutch and at UW, when patients come into us, we have a chance to, put on PPE and get really protected and feel pretty safe. And early in COVID, as a first responder, you didn't really know what you were getting into back then. Tell me a little bit about how you sort of approached that. I mean, what was it like those first couple of weeks when, we didn't know how COVID spread. We didn't know whether it was droplets or coughing or in the air or body fluids or whatever it was. What was it like going on calls then and going to have to help patients not knowing really much about the virus at all?
Sure. Well, it's interesting as far as responding to patients in the field. The information was out from King County EMS very clear after the first Snohomish County case, where full meg, mask, eyes, gloves, goggles for suspected COVID patient. But that just protected us at the call. That didn't protect us at the station. That didn't protect us at the crew level, because we still went back to work and cohabitated like we would before and after the pandemic. So it was the first time as a first responder where you could actually just show up to work and be a risk to your crew. And that was an interesting challenge to face. Nobody wanted to, not too many people were worried, but there were a lot of people worried about being the super spreader, being the one to take down the station, being the one to check and not know it. And there wasn't testing. There was only isolation at the time. And as people started to go into isolation, your heart broke for them because you wish they could get tested and come out. But that wasn't available. So being part of this study was my first COVID test. And my first opportunity to have some information about myself to make sure I wasn't a risk to the people that I was around at work.
That's a great point, and a risk to your loved ones and other people in your life as well, which is pretty amazing. Julie, a couple of comments about communication during the pandemic, and it's been hard to know what kind of information we can rely upon, and how do we trust various different findings that come out. How have you advised people and participants in the study, what to pay attention to, what to ignore? I myself am a physician and a scientist at the Hutch, and I struggle immensely with knowing what's news and what's fake news, and trying to sift through information and make decisions in real time. How do you advise people, particularly with COVID, to make those decisions?
That's been a really big change being a healthcare provider. You learn about the disease, and then you're able to say, these are the facts. But the facts of COVID are constantly changing. And so it was the first time in my career that I had to say, I don't know, all day long. We don't know what's happening. And so what we've done as a study is we take the facts that are based on research that come from trials that have been conducted, coming out of labs or clinical groups. We just say, this is what we know. This is what we know today. And what we know today may be different tomorrow. In the beginning, when Mike, got his vaccine way back at the start, we're like, okay, you're vaccinated. You're great. Well, now we know you're not necessarily protected against COVID if you've been vaccinated. But so the story, has changed, and we've had to be more willing to say, we don't know readily, and also to just get out that information as fast as possible. We're not waiting for everything to be published. We're like, this is what we know now. This is, how we're going forward, and that may be different in the next month. And Mike has always been right there. What do you know this time? And willing to go with the flow.
And so the last question, Mike, is any advice you have for people who are thinking about participating in studies at the Hutch and University of Washington? Meaning, we exist because of volunteers, because of people who trust us and decide to participate in studies. There would be no Hutch, there would be no UW Medicine if we didn't have that. Any advice to people thinking about it?
Sure. Well, for me, it was right time, right moment. My curiosity was there, my availability were there, and the messaging was there, and it was relevant to what we were doing. So if the message is in front of people at the right moment, I think you'll have a lot of volunteers come in for more studies.
Terrific. Well, Julie and Mike, thank you so much. You'll be back for the questions, I know, because the questions are starting to roll in. I want to just remind everyone, please put your Q&A questions into the queue, and we will ask them during the Q&A section. I now want to bring our third group in today, and this is Rhonda and Makia. So Rhonda Reese and Makia Mansell. Rhonda is a genetics researcher in Sohail Mosinski's lab, studying childhood leukemia, and Makia is a junior at the University of Washington, where she's studying molecular and cellular biology and bioethics. They met in 2019 after Makia's junior year of high school, when she participated in our summer high school internship program, otherwise known as the SHIP program, which incidentally is funded by donors of the Hutch. We very much appreciated this, which were one of the programs we're most appreciative of and proud of. The project they started together that summer has blossomed into really promising experimental treatments for children with leukemia. Makia is still working in the lab part-time and headed toward a career in medicine and cancer research. So welcome, Rhonda and Makia. Terrific to have you here today. And I thank you so much. You're both so incredibly busy. Thank you so much for being here. So I guess my first question, Rhonda, are you there? I don't see you on the queue. Can you just say if you're there or not?
I am here. I don't see my video, but.
Okay, well, you're there by voice, which is terrific. So I'll ask you the first question. As a scientific mentor, when you meet someone like Makia, what are you thinking about?
Yeah, we love to participate in the SHIP program. The lab has probably been participating for about 10 years now. And our goal for them is to really showcase for them what a career in research science looks like. And that means giving them a project that they can call their own, that they can really, with our guidance, kind of oversee. And that way they get to see the good, the bad, the ugly of what scientific research really can be. We guide them through the troubleshooting process. We guide them through changing variables when invariably an experiment doesn't work. We guide them through all of that. And we hope that in the process, we are also showing them our passion for, what we're doing, which is, looking for cures for childhood leukemia. And Makia has certainly, she's been an enthusiastic participant in that program, and she has really embraced the research that the lab has done and really been an integral part of not only the program, but follow up as a participant in our lab.
So before I ask the question to Makia, first, one giant apology for calling you Makia. My apologies. That's not acceptable, and I'm really sorry. Okay, so
no worries. Thank you. I appreciate that.
So Makia, you've accomplished a ton, and you're only a junior in college. Okay, you could do anything. You could have gone. I mean, Microsoft is sponsoring this. You could have gone and been a programmer in Redmond. Okay, and written Microsoft programs. You could go work for Costco and decide how to market and distribute products. And you go to Starbucks. You go to any Seattle great organization. But what made you? What excited you about science, and what made you sign up for the SHIP program? And tell me how you think about yourself and your scientific career.
Yeah, so I guess my experience with SHIP was, somewhat of an accident. I mean, I of course applied intentionally, but at the time it was a program that kind of felt bigger than me. The Hutch is such a cool place. It's had a big impact in my family. My grandma was diagnosed, 12, 13 years ago now with stage 3 multiple myeloma. And, I'd heard about the Hutch. I'd happened to visit a couple times with my grandparents. And I was kind of referred or suggested to apply for the program by another mentor. And I threw my hat in the ring thinking, it'll be a cool thing to have tried to go for. And then, sure enough, I got the spot, ended up in Sohail's lab working with Rhonda and a few other people. And, 3 years later, I'm still here. And I think it's been a great way to kind of, like Rhonda said, see the good and the bad and the ugly about science, where things are maybe a little bit more difficult than you're expecting, and how to just keep working to find those answers to, really hard questions. And I think moving forward, I'm definitely going to be taking away a lot from, the last 3 years of my experience at the Hutch, as the big picture of this conference, I guess, collaboration, and just how important it is to work with other scientists and work towards a common goal.
Terrific. And I just want you to know, Makia, that recruiting starts today, okay? We want to talk to you in about 12 and a half years, okay, about coming back to the Hutch for a scientific job. And it'll take you about 12 and a half years to get there, but we've got one for you. So keep us in mind, okay?
Got it. I'll mark it on my calendar.
Mark it on your calendar. So Rhonda and Makia, both of you, could tell us a little about the project you're doing in Sohail's lab. So really, Project Stella is really incredibly interesting to me. Try to share some of that excitement with the audience.
Sure. So actually, the summer that Makaya volunteered in the lab, we had a project for her and her partner, but there's a lot of downtime, especially when you only have a single project to work on. And so in the downtime, we asked these students to build us a database of existing drugs that are already in clinical trials. And so we asked them to go to clinicaltrials.gov, the website, and entering a few key search words. We asked them to basically copy and paste, pharmaceutical companies, the drug name that they produced, the gene target that drug works on, and then the phase of the trial that was in progress. At the time, there was really no way to download that information, and since they had some time on their hands, we asked them to help us out by doing this copy-paste, build this spreadsheet and database of genes and the drug targets. And through that effort, we really were able to match up gene targets with highly expressed genes in these leukemic patients, and really try to find alternative therapies and more personalized medicine approaches for these patients that really don't respond to standard chemotherapy regimens. And so Project Stella is a great example of what happened from that. So a little girl named Stella Novotny, she had a very rare and highly aggressive form of leukemia that really affects children less than 5 years of age. And Stella's family reached out to Sohail. He's a high-risk leukemia expert. And throughout the course of her treatment, they were in close contact with Sohail, and he was able to get them some compassionate use therapies, which unfortunately Stella's disease was so advanced they didn't work for her, and Stella ultimately succumbed to the disease. But her family, really championed the effort to kind of push the effort and the research by fundraising with their friends and family, and they created this program called Project Stella here at the Hutch. They raised a significant amount of money for the lab, and that enabled us to really take these. We found a target for this highly aggressive form of leukemia that Stella had. It's a gene called folate receptor. There's a drug that already exists by a company called Sutro. We were able to obtain that drug and test it. We built models in the lab to test that drug, and it effectively killed the leukemic cells. And the mouse models that we created for it, it effectively eradicated the tumors in these mice. And so really the effort that they started has really become a result of really the project that Micaiah had when she was in the lab, building this database of drugs, what we call the drug repurposing project, which is repurposing drugs that are already out there and trying them for a new indication. And so as a result of Project Stella and the program and this philanthropy that this family and other families have recently joined the effort, we now also built a CAR T to direct towards folate receptor, and that clinical trial will start probably early in 2023 and will be the first philanthropically funded clinical trial here at the Hutch. So we're really proud of that.
And it kind of has to be, given how rare the disease is. Micaiah, any additional comments about how this project has impacted you?
I think it's given me a very realistic impression of science and kind of how long it takes. I think, like I've said a couple times now, it's been 3 years and I've kind of worked off and on this project. And, seeing just how far Project Stella has come has been truly incredible. I believe it or not, I actually remember kind of like poking around at various drugs that I was copying and pasting into this database, and different aspects would pique my interest. And I kind of remember kind of copying and pasting the Sutro drug, and now we're here. But yeah, I think Project Stella is, it's a really cool project to talk about. It's very much kind of like grassroots and like funded by families that have been impacted. And it's really just a very important project and very, very heartwarming, the whole story.
So first of all, I want to really thank you for being at the Hutch and UW, because together those are just two spectacular institutions, and we are so lucky to have both of you in our midst. So thank you for being here, and thank you for all you're doing to advance the cause of rare leukemias. I actually had the chance to meet with a family whose granddaughter has a rare leukemia like Stella, and they were just touched and passionate about the work of Project Stella. So keep up the great work, and we'll be back for the questions with you in just a few seconds. So before the questions, I just want to say, just reflecting about that discussion I had with the family who has a grandchild who has a rare leukemia. We do so well for most children with leukemia, but these rare forms of acute myelogenous leukemia in young children, infants and toddlers, are extremely hard to treat. And the grandfather could not get through the session with me without breaking down and just being overwhelmed with what his 2-year-old granddaughter is going through. And again, the research is what makes the difference. It's the research that fuels life-saving treatments. And so just to give you an example of what some of the philanthropy that you people on this call have supported, funds to support the high school internship program that Makia attended, a really important program here at the Hutch, which has been ongoing for 20 years, has made a huge difference to what we're doing. Your funding enabled us to create the COVID-19 Clinical Research Center so that Julie and others can safely conduct studies of vaccines and treatments and antibodies. And it's for COVID now, but who knows what virus is going to be next. And we're going to need to have that facility available. And philanthropy has made an enormous difference in Sarah and Cyrus's breast cancer metastasis research, how they treat patients. So there are many, many ways to contribute to the Hutch. One of them you saw was the importance of volunteering for a research study. That's incredibly important. Second, keep attending Science Says and register for events like Obliteride. Third is make a donation, and some people can make an enormous donation and some people can make a small donation, but the act of giving is something which is incredibly important. I know it's important for me and my family. It's important for you and your family to participate, and it can be an incredibly rewarding experience, and it's give as you can. And there's some years that people can give a lot. There's some years you can't, but you can give something, and so we appreciate everything you can do of any size to the Hutch. Please explore our science education website for more information about some of the science programs that we do, and share what you know about this place through social media when you're on Instagram and Twitter and Facebook and other areas like that. This all makes a difference. If you're thinking about making a donation today, I'm thrilled to share that for today, John DeLoe, a longtime supporter of Fred Hutch, is matching gifts we receive today up to $100,000. Another example of the power of partnerships. Last year, we raised $682,000 on Giving Tuesday. Let's work together to get us there this year and try to do it again. And if you're inspired to donate, please think about donating before midnight, and we can apply that to the matching gift. And you see today on the screen, you can see the URL to go to and scan the code, and you can make a donation that way. So thank you. I'd now like to bring our panelists back for our Q&A. I'd like to start off by just thanking everyone for being here and helping us talk about the incredible things that happen at the Fred Hutch. And the first question I'm going to ask comes from Farida Elliott, and it's a question I have great interest in. And Julie, you might be in perfect position to answer this one, which is, what is the study, since you were following people early in the process, what has the study told us about people who never get COVID? And what about that patient population? What do we know, or do they not exist and we all get it in some way? What do you think, Julie?
Well, I'm not sure yet, because every week right now, new people in our study who haven't had COVID this whole time are getting COVID. So maybe you just haven't met your virus yet. I'm not sure. And then the other thing is that we do see people who have had COVID, clearly have had COVID. We can see it in their blood, but they had no idea. So maybe the subclinical infection is where a lot of people are, and we're just not doing that mass testing to know that. So I'm not sure yet if people are really not getting COVID. I think there probably are definitely people who have some kind of genetic diversity and they're not able to get infected. But I think more likely we're just still seeing people getting COVID that just haven't had an opportunity to get it yet.
And so, Mike, I guess I'll ask you a similar question. One is, when you did this, did other first responders from Eastside join in the process? Did you do it alone? Was it onerous? Were there a lot of procedures you had to go through? What was it like from the participants' perspective?
Sure. Thanks for asking. Due to HIPAA, I'm not allowed to answer a lot of these questions. That's what Julie would tell me when we would come in and be like, hey, you saw my friend earlier this morning.
Yeah, you can't say that, right? Exactly.
Yeah, Julie and her team were so wonderful throughout this whole process. Caring, compassionate, consistent, and it was very evident amongst some of my coworkers, who had the opportunity to meet Julie, because we talk about Julie and the study a lot at work. So yes, several people. I was able to recruit some and bring some in, and several other coworkers. We organically found our way to talking about Julie and Fred Hutch and being part of the study.
Terrific. Thanks, Mike. So Diane asked a question, and this is for Sarah. So Sarah, if we know that a pretty significant percentage of early stage breast cancer patients go on to develop metastatic disease, and you're the one who's seeing the early stage patients, it's your practice, you're seeing the women with the small breast cancers or the locally advanced breast cancers, what do you do to find out if they've got metastatic disease at presentation? What is the staging like? Do you see a role down the road for more innovations in staging? And then lastly, what about molecular staging? What about doing blood tests looking for genomic profiles in the blood? There are a couple of companies now that are trying to advance this process. Tell me a little bit about what you think about how we profile early stage breast cancer.
So our current society guidelines like ASCO or American Society of Clinical Oncology and National Comprehensive Cancer Network do not recommend distant metastatic staging, so body imaging like PET-CT or CT scans or bone scan for early stage breast cancer defined as stage 1 or 2. For stage 3 and beyond, 3, yes, we do, or based on symptoms. So if a woman presents with new back pain and associate, at the same time she has a fairly large tumor in her breast on exam, even if we don't have clear evidence on imaging of metastatic disease in the axilla, we might consider a staging workup. But in terms of monitoring and surveillance for metastatic disease, there really is, with regards to blood markers, the tumor markers like CA19, 19 and CEA have not been shown to be predictive, and there's a low sensitivity and specificity in terms of monitoring and looking for metastatic disease recurrence. So they're not utilized in clinical practice, nor are they recommended. And in fact, in terms of surveillance, someone asked in the chat too about imaging and the role of imaging for early detection of recurrence. There have been 2 large randomized control research trials examining the efficacy or utility of imaging, like CT scans of the chest, like your patient with a pleural effusion, and monitoring regularly with imaging. There's been no proven benefit either in overall survival or disease-specific survival when you image regularly versus just do yearly physical exams and surveillance mammography. So we don't do regular imaging looking for disease because it doesn't change the outcomes of women with breast cancer.
Terrific. Thank you, Sarah. Now there's a question which actually is going to bring together and create a new collaboration on this call, okay, which is Mike and Sarah, okay? So Mike thinks, God, he's in the COVID group. How could he be involved in the breast cancer group? So the question that's come in from Rachel to Dr. Javed, it says, you're asking people to agree to a bone marrow biopsy for a study that won't necessarily benefit them. What's your pitch, and why do patients want to participate? And Mike, since you've already participated in a study, I'd like you to approach it from the same perspective of being the potential patient. I know it's not likely, but how would you look at that? So Sarah first, and then Mike.
Thanks. That's something we just recently talked about, Cyrus and myself, as we recruit, up to 900 patients for this upcoming study. You'd be surprised how giving people are when it comes to wanting to help advance the disease and their peers. Everyone knows someone who's been affected by breast cancer, siblings, family members, friends. And so I just spoke with a patient yesterday, in fact, and consented her for the study. She knows that it's not going to help her directly, but she wants to help out and further the field and help others in the future. So I think a lot of our patients, most women, but some men, I'm seeing a man today, are really receptive to research and advancing our field and helping others.
And Mike, what is it like from the volunteer perspective when you're being asked to do a voluntary bone marrow biopsy?
So I think I would have to start with trust, and then a belief that your instilled trust is better for somebody in the community or the greater community in general. I would not just give anyone my bone marrow. If Fred Hutch asked me to come in and give them my bone marrow, I would consider it. So it's having that ability to be trustworthy and caring and compassionate throughout the study process that makes it something worth considering.
And I think that's a really, really, really great point. And my next question is for Makia and Rhonda. So the study you're doing with Project Stella, and so much of what we're doing now in cancer and viral research focuses on data, okay? And I'm really curious about how the two of you view each other, view yourselves as data scientists. And as we enter this world of data, what I can tell you is that my generation are nervous about data, okay? Once you get down to Sarah and Cyrus's generation, they're a little more comfortable with data, but they're not completely comfortable with data. Makia, I think one of the things that you're going to bring to the table is that your generation's going to just be natural and really comfortable with data. Please confirm that, or tell me I'm crazy, okay?
I think being a part of a generation that, grew up with ready access to the internet, you're used to, going on maybe a Google deep dive on some crazy story you heard. And I think that does carry over to data and what you're passionate about, what you're passionate about learning. And I think, like I mentioned earlier, I would, if something piqued my interest in a study I was looking at, I would, kind of go down a little rabbit hole and look into different aspects of that study or that just subject in general. And I think maybe older generations aren't as comfortable with data, but I think it's a matter of going and exploring and kind of sitting with something that maybe isn't as comfortable. A headline that maybe doesn't sound quite right, go and look at it. Find a couple papers, go and peruse a few sources. I think data is a really cool thing. I think you just have to really know how to look at it and work with it.
Thank you. That's fantastic. And Rhonda, how are you thinking about data science and how it's impacted your career?
Yeah, I mean, I would consider myself a data scientist at this point. I mean, I rarely work at the bench anymore, and it's really been driven by, when you work on a very rare disease, you need to have the opportunity to work with cooperative groups to bring those samples together. Sohail is part of the Children's Oncology Group, which is a national organization that oversees clinical trials. And one opportunity that those participants have is to donate their samples for research. It's through that collaborative process that we've been able to sequence, close to 2,500 patients with childhood leukemia of all various subtypes. When you're talking about a really rare subtype like what Stella and Ella have, out of 2,500 patients, there might be 50 patients that have that same subtype. And so data science is really important, especially for very rare diseases where, when you have the opportunity to aggregate a large cohort, even if they have the umbrella term of AML, there's a lot of different nuances to the different types of AML, and I'm sure this is true for other diseases. And so data science becomes really important to be able to not only harness the power of enhancing the numbers of those rare subtypes within a large cohort, but also to be able to compare those subtypes to each other and see what are the shared properties and what are the different properties. And data science really helps us to do that. And so, folks like Micaiah, every year I'm just amazed at the students that come in the lab and already know how to program in languages like R and Python. And I encourage them, I say, keep that up, because if you pursue science as a profession, you're going to be so much better at being able to understand the data that you generate, if you have the ability to kind of programmatically manipulate large data sets like what we have. So yeah, data science is where it's at for me.
It really is. And we're incredibly proud of the data science groups we have at the Hutch. Jeff Leake being our new chief data officer, Rob Bradley running our data science IRC, both making a huge impact. One of the things Rob has really influenced me is the idea that he talks about the democratization of data science. And we all have to feel we're data scientists or we're not going to be successful in what we're trying to do. Cyrus, can you just comment on that in your laboratory and how that's impacted the way you approach science?
Yeah, I mean, we've always been very married to the bench. We're cell biologists really by and large, but at some point you do have to evolve. And if you're not evolving, you're staying still, you're not pushing boundaries. And so I think the best example of this is, already the fruits of the study that Sarah and I are working on together, along with Chris Lee, along with others. We're purifying disseminated tumor cells from bone marrow. We're getting single cell transcriptomes of these cells. We want to find targets on their surface that we can target with immunotherapies in order to get rid of them. But we need to know what's specifically on their surface that's on no other cell surface in our body. It can't be in the esophagus. It can't be in our brain. It can't be in our lung. How are we going to do that? Well, the Chan Zuckerberg Biohub just published a data set of 500,000 cells from 24 different organs. Somehow it didn't have brain in it. Allen Institute just published a data set of brain, 1 million cells, single cell profiles of brain cells. So we have a map now of like 1.5 million cells from the human body. We can actually layer on the disseminated tumor cells we profile onto that universe, do differential expression, figure out what's unique to the surface of these cells. We already have a few targets just off of the first few patients that we've enrolled on the study, and this is going to grow with each of the patients that we profile. And so, that is big data. It takes people not like me to do it. So we have to bring in computational folks into the lab, and we do. We have a fantastic one. And it's really, really fun to explore data in this way and to work with programmers, people like Micaiah who actually know what they're doing on a computer.
Terrific. And the last question is going to be from Mike and Julie, and we have 2 minutes, which is good. And it's a really good question from Rachel, and she asks, for Julie and Mike, did you have study volunteers or colleagues who were skeptical of vaccines? And if so, how did it affect your collaboration?
Go ahead, Mike.
Okay. Absolutely. The participation rate among first responders prior to the vaccine mandate was not as high as the general population. at least speaking for the King County numbers. And so there was, and there is, and remains vaccine speculation, absolutely. Where we are now is we've evolved from a year of a vaccine mandate into considering the benefits of the vaccine and what that actually means to be a responder. And each agency is having to approach it very differently and very delicately, because we're talking about people who are passionate about being first responders who who have either a belief or a legitimate or a religious exemption and want to come back to work. So we're still living that very, very right now and trying to get through that.
Great.
Yeah, and we have participants in the study who still have chosen not to be vaccinated, and we respect that. And we just present the science that we have, and we talk about the science, and it really hasn't been a problem. I think that people that are coming here to participate in research, they care about research, and we can respect that. And so we still work well together.
Julie, thank you. And because you guys were so quick in your answer, we can squeeze one more in, okay? And this one's for Sarah. And it's a great question. Do all NED patients have dormant cancer cells?
I don't know the answer to that. We don't monitor or check for that. I know it's been increasingly popularized in the lay press, the value of these, but it's still not ready for prime time usage. We know that even, 2/3 of women who do have circulating tumor cells in their bone marrow do not go on to develop metastatic disease. So we don't want to overly alarm people or overtreat people with these found incidentally.
Terrific. Thank you all. I want to thank, this was, I do these all the time, and I got to tell you, this was like, I shouldn't say it's my favorite because other people will think theirs should have been the favorite, but this was a great one. You guys are absolutely fantastic. Really interesting examples of collaboration. So thank you all for being here today, and thank you all for what you do to the Hutch. And to those who've joined us, if you're interested in the science you heard today and you want to learn more, visit us at fredhutch.org to learn. And again, people like you fuel our research and the educational programs like SHIP. Please consider making a gift to the Fred Hutch today at fredhutch.org. And remember that John DeLoe will match every dollar you give today up to $100,000. Our next Science Says will be in February of 2023. Look forward to seeing you all there. Thank you so much and have a fabulous December. Bye-bye.